Okay, next we have Alexandria Hughes, a neuroscience graduate student at University of Colorado. Alexandria attended Boise State University, where she received bachelor's degree in biology and worked on research projects investigating unfolded protein response signaling. After that, she began her doctoral training at University of Colorado as a PhD candidate in neuroscience. She joined the lab of Dr. Bruce Appel, where she is currently studying how myelin sheath formation and elimination are being regulated in the central nervous system. During her time in grad school, Alexandria received numerous awards and funding, including NSF Graduate Research Fellowship, and she also published several exciting papers. And she will discuss her most recent work in Nature Neuroscience entitled Microglia-Phagocytose, Myelin Sheaths to Modify Developmental Myelination. And I'll give it to Alex.
Thank you. All right. So thanks so much for that introduction. I'm excited to be here with you all. This is very cool. All right, let's get into it. Maybe. So in the central nervous system, a glial cell type called the oligodendrocyte wraps neuronal axons with myelin to increase conduction velocity and provide trophic support to axons. This is an individual oligodendrocyte that I've labeled in the spinal cord of a zebrafish. Its cell body is here, and these rectangular structures are myelin sheaths, extensions of the plasma membrane that wrap compactly around axons to form individual units of insulation. It used to be thought that after these myelin sheaths were formed, they were static throughout life. But in recent years, we've learned that different neuronal activity paradigms can change myelination. Myelin sheaths can grow and elongate. They can also shrink or be lost altogether, all with potential effects on conduction velocity and higher brain functions. But how myelin sheaths change is not well understood. In the first part of my PhD work, I investigated mechanisms underlying myelin sheath growth. And we found that this process is mechanistically similar to synapse formation in a lot of ways. Axons accumulate synaptic vesicles and exocytose them under nascent sheaths, which harbor postsynaptic scaffolding molecules that you may know from synapses that anchor transsynaptic adhesion molecules that, together with the axon, promote the elongation of these sheaths. So we were able to learn a lot about myelin sheath growth by comparing this process to synapse formation. But there's another major similarity between myelin sheaths and synapses that we didn't investigate at all in this work. And that's that following robust formation during development, both myelin sheaths and synapses undergo a period of elimination. These are time-lapse imaging data from zebrafish oligodendrocytes from Chang He's group in Shanghai, where individual oligodendrocytes were time-lapse imaged, and myelin sheaths that persisted or stuck around are marked with these green arrowheads, whereas these sheaths that are marked with red arrowheads shrunk or disappeared altogether during the 40 hours of imaging. And altogether, the group found that about 1/3 of all myelin sheaths that are formed are spontaneously eliminated afterwards. But the mechanism underlying the disappearance of these myelin sheaths is not known. But myelin sheaths are not the only structure in the developing nervous system that spontaneously disappears. We know that synapses, neuronal precursors, and neurons are all overproduced and then pruned. And in these cases, these structures can be eliminated by another glial cell type called microglia, shown here in blue. And so because myelin sheaths disappear and we don't know why, we raise the big question of whether myelin could also be pruned by microglia. So to begin to think about this question, it's helpful to think about what we know about microglia elimination of these other structures like synapses. We know from work from Dorothy Schaefer when she was a postdoc with Beth Stevens that microglia eliminate synapses in an activity-regulated manner. Dorothy investigated synapses formed by retinal ganglion cell axons toward the lateral geniculate nucleus in the mouse, and she found that if she silenced those axons with tetrodotoxin to block sodium channels, microglia engulfed more synapses than normal. So what do we know about myelin and activity? We know from work from David Lyon's lab, as well as Jake Hines, a previous postdoc in our lab, that when neuronal activity is suppressed by a slightly different method, this is tetanus neurotoxin, oligodendrocytes form fewer sheaths, and sheaths that are formed have a shorter lifetime before spontaneously disappearing. So taken together with the observation that microglia eliminate more synapses when neuronal activity is blocked, is it possible that these myelin sheaths are being eliminated by microglia more robustly when neuronal activity is blocked in these paradigms? So taken together, we hypothesized that microglia phagocytose myelin sheaths in a neuronal activity regulated manner, similar to their elimination of synapses. And from this hypothesis, we can form a few predictions. Of course, this is unlikely to happen if microglia never interact with or phagocytose myelin. We would predict that these interactions would be scalable with neuronal activity, and that if this program matters for the development of myelination, then perhaps blocking it by eliminating microglia, we might predict an excess of myelin to develop and persist in the nervous system. But to ask these sorts of questions, we need to be able to investigate all of these cells during normal development. So we use the zebrafish as a model system to do this. Zebrafish are genetically tractable. We can inject plasmids and things at the one cell stage to label different cell populations. And then we can perform time-lapse imaging because they are optically transparent. And so everything that I'm going to be showing you today takes place at this 96-hour or 4-day time point in which myelin sheaths are being formed and eliminated, and microglia are present in the central nervous system, having already invaded as macrophages and turned into microglia. So unless I say otherwise, we're going to be looking at this time period. All right, so do microglia interact with the oligodendrocytes? To ask this question, we needed a way to look at microglia. So I generated a transgenic line in which regulatory DNA from a gene that's expressed in macrophages and microglia drives expression of a membrane-tethered yellow fluorescent protein called mVenus in these cell types. And so you can see in the spinal cord of a zebrafish, if we line up several on an imaging dish, you can just image over the spinal cord. We can see microglia in the spinal cord, and crossing this reporter line to one that labels myelinating cells show us the major myelinating tracts in the spinal cord. And over the course of development, there's a couple things I want you to notice. Microglia are present in the myelin tracts, first more numerous in the ventral tract and slowly accumulating in the dorsal tract. But we almost only see them within these myelin tracts. We hardly see them in the void space in between, raising the possibility that the microglia are specifically attracted to the presence of oligodendrocytes in these tracts. And so we counted oligodendrocytes using a different reporter line for oligodendrocyte cell bodies. And we found that both microglia in yellow and oligodendrocytes in magenta distributed similarly within the dorsal and ventral tracts over time, at least indicating that these cell types are in the right time and place to interact with each other more specifically. So to look at these interactions directly, let's zoom in on just one microglial cell. This is a single microglia in the dorsal tract of a 4-day-old fish. Its cell body is here, and these processes extend out. And these magenta rectangles are myelin sheaths. We see that most of the microglial processes course out along the myelin sheaths, sometimes even wrapping around the outside of myelin sheaths, and sometimes just reaching out toward potentially unlabeled targets. But by looking at a number of these microglial cells and plotting the fraction of processes contacting these different types of structures, we find that the majority of microglia devote the majority of their processes to contacting myelin sheaths, only seldom contacting unlabeled targets such as this or oligodendrocyte cell bodies. So this specific contact with sheaths suggests a specific interaction, but this is also just a still photo. Microglia are actually quite motile. Over about 3 hours of imaging, we can see this cell is just examining all of this nearby territory in the dorsal spinal cord. If we take it out even longer, we see that they just pass through the area every once in a while. This is over 13 hours of development. So microglia seem to be specifically contacting myelin and contacting a lot of myelin over time. But are those microglia actually phagocytosing myelin? Because microglia are not always phagocytic. And classically, microglia state is inferred from morphology. Sometimes you'll hear microglia referred to as ramified or amoeboid, referring to these different morphological states. And it's thought that only microglia that look like this are participating in phagocytosis. But in any disease state, you can find microglia along this spectrum, so it's not perfect. But another measure of phagocytic activity is the presence of intracellular calcium transients in the microglia. This was first found in the early 90s, where macrophages were loaded with the ratiometric calcium indicator diaphora-2, and then monitored as they engulfed labeled beads. And the group found that macrophages exhibit a calcium transient just prior to the onset of phagocytosis of the beads, and then that drops, allowing phagocytosis to occur. And later work from Mario Capecchi's lab, for instance, has shown that upon laser injury or other things that cause microglia to phagocytose, the general frequency of calcium transients is a marker of phagocytic activity. So to look at calcium transients in our own system, I generated another transgenic line to label calcium in microglia. This is an individual microglial cell in the ventral spinal cord. Cell body is here, and these processes that are just lashing out within that ventral myelinated tract. And you can see that this cell exhibits a number of calcium transients labeled by the genetically encoded calcium indicator GCAM6S, here fused to the membrane. And this is over about 10 minutes of imaging, and this cell exhibits a number of these. But there is a particular challenge to studying calcium in microglia compared to imaging calcium in neurons, for example, and that's because these cells move. And so typical ROI-based measurements of calcium don't work. The cell is also moving, so just as a function of membrane moving, you don't always know if the change in fluorescence is due to the actual biosensor detecting calcium or if it's just addition of membrane. So to differentiate between these things, we did two things. We generated fish in which calcium was labeled in microglia along with just a membrane tag so that we could distinguish events that are just membrane fluctuations from those that are true calcium events. And we also analyzed calcium events with this great calcium indicator toolbox called Aqua, developed by the Poscanzer and U labs recently last year, to detect calcium and membrane events within these microglial cells. And we found that on the basis of intensity, we were able to separate just membrane movement from actual calcium events, therefore allowing us to study calcium events in microglia without the confound of membrane movement. So now this is great, but all we really care about is calcium events when we're looking at microglia interacting with myelin, so let's get to it. This is a microglial cell expressing that same calcium indicator. Here filtered to remove just membrane events. And in magenta, we have the same myelin sheaths that I showed you before. And you can see that this cell is interacting with a number of myelin sheaths exhibiting transients detected by Aqua within processes that are contacting sheaths and those that are not contacting sheaths. And if you look closely, you can see some myelin jumbling around the inside of it as well. And we found that a number of these events occurred specifically at contacts with myelin sheaths compared to processes that are not contacting myelin sheaths, potentially indicating a specific interaction with those sheaths. And we were delighted to find examples where high intensity calcium events were followed by blebbing and selective loss of fluorescence in the underlying myelin, suggesting phagocytosis in line with that thing that I showed you from the early 90s in macrophages. So microglia appear to be phagocytosing myelin, at least in this paradigm. But to track this more thoroughly, we performed longer time-lapse imaging with that M. venus line that I showed you before. And we also found phagocytosis of sheaths. So in this video, a nascent oligodendrocytes, brand new, it's starting to form myelin. You can see in these rectangular boxes just a couple of nascent sheaths that are soon engulfed by this microglial cell. And if you watch this over the next couple hours, the cell continues to try and make myelin sheaths, and these attempts are just thwarted by this microglia. But this isn't just the case for brand new myelin. We also saw this for more mature myelin made by oligodendrocytes that transcriptionally activate the major myelin protein MBP. where a microglial cell extends around a myelin sheath and slowly dissolves it within the microglia. So microglia appear to be eliminating myelin sheaths, but previously it was thought that myelin sheaths simply disappeared on their own, independent of microglial contact. And this process was called retraction or membrane resorption. And so we were interested to know how many myelin sheaths microglia eliminate in the developing nervous system between microglia engulfment and cell autonomous retraction of sheaths. So to compare these two processes, we performed time-lapse imaging of microglia interacting with myelin, and then processed successive time-lapse frames in such a way that we detect change over time. Here that's plotted in cyan. And this effectively reconstructs sheaths that have disappeared during the imaging scheme. And so this allows us to identify all sheaths that disappeared. And then by looking at the video, we can determine whether those were engulfed by microglia, like this example, or if they disappeared or changed on their own, independent of microglia contact. We might call that retraction. We found that the majority of sheaths that were eliminated specifically by microglia engulfment, although sheaths did sometimes disappear on their own. But microglia engulfment accounted for about 70% of all missing sheaths at this stage, suggesting that this is a major mechanism of myelin sheath refinement in the developing nervous system. It's important to note that both populations of sheaths, those that were engulfed and those that were retracted, are both much shorter than the average sheath. A normal sheath is about 30 microns long, and both of these are between 5 and 10 microns long. But those that were engulfed were longer than those that were retracted, potentially explaining why both of these mechanisms might be necessary. Perhaps engulfed sheaths are more mature, for example. But we now know that most sheaths that disappear are engulfed by microglia, but it's not known if all microglia participate in this process or if this is secondary to their other functions in the developing nervous system. So to figure out how widespread microglia phagocytose is among microglia, we needed a way to label fluorescent myelin within microglia. So the problem with this is that our fluorescent myelin disappears. It loses its fluorescence when it's engulfed. And this is due in part to the acidification of nascent phagosomes that take up that fluorescent material. So we reasoned that inhibiting the proton pumps that acidify these phagosomes might prolong the fluorescence of myelin within the microglia and allow us to study how widespread this phenomenon is among the whole microglial population. And so this proton pump is druggable with baflomycin, a drug that blocks it. And so we treated larvae carrying transgenes for microglia and myelin with either baflomycin or a vehicle control, and then looked at myelin inclusions within microglia. And we found that when we treated larvae with baflomycin acutely, we could see these bright myelin inclusions within microglia. And we can still see myelin inclusions when we treat larvae with DMSO, but they're a little bit fainter, smaller overall. But to study these myelin inclusions in more depth, we used a morphological segmentation pipeline to identify these inclusions while excluding other material that's passing through the field of view. And then we can get all sorts of information about them. And perhaps the most obvious thing we see is that there are more inclusions overall when we treat with baflomycin. Consistent with stalled breakdown, now we're seeing more of these inclusions. But it's not really the most interesting thing we can learn. Instead, what we found was that all microglia that were treated with baflomycin contained some number of myelin puncta. We never detected any microglia without them, indicating that all microglia in the spinal cord seem to be participating in myelin phagocytosis. All right, so microglia interact with and phagocytose myelin. But if you remember from the time-lapse imaging videos that I showed you, for example, microglia manage to contact quite a bit of myelin without eating it, implying some selectivity to this process. And so we hypothesized that neuronal activity regulates the phagocytosis of myelin by microglia. And to investigate this, we left the spinal cord and went to another brain region where there are more defined paradigms for manipulating neuronal activity. This is the optic tectum, the visual processing center of the zebrafish brain, homologous to the mammalian superior colliculus. It has two defined regions. Shown here in green are neuronal cell bodies. So there's this cell body layer of the optic tectum and a neuropil layer. This is important because microglia only really occupy the cell body layer. So here in cyan are cell bodies that belong to this layer, and this asterisk marks the neuropil, where axons and dendrites from these neurons are present, but you can't really see them because this is a cytosolic label. But what you can see are that microglia extend within the cell body layer. And if you were to take the fish and now look down on one tectal hemisphere, you'd see that these microglia, again, absent from the neuropil, are lined up against this magenta structure. And this is the tectal commissure. It is the only source of myelin for these microglia to eat during this, in this region. And so just to orient you, in this rotation, we have the tectal commissure labeled here in magenta. And these two large magenta structures are the neuropil, labeled by this transgenic line. But you can see the microglia are large cells, that fill out the entire neuronal cell body layer. And they're positioned in between that tectal commissure, now here alone in magenta, and these neuronal cell bodies present in the cell body layer. Now, previous work from Zhu and Du's group in Shanghai showed that microglia are drawn to contact these cell bodies when these neurons are active. So here's a picture of some of those contacts that can be formed between microglia and those cell bodies. And you'll see these microglia do contain a little bit of phagocytosed myelin, raising the possibility that microglia that are out contacting these active cell bodies might be less likely to phagocytose myelin from the commissure, just as a function of position. If the microglial cell is over here, because these neurons are active, it might be less likely to phagocytose myelin over here. And so to test this hypothesis that active neurons delay or prevent microglia from phagocytosing myelin from the commissure, we're going to use a variety of manipulations to change activity in this region, and we'll measure both microglia neuron contacts and myelin phagocytosis from the commissure. So first, we wanted to inhibit activity of the cell body layer neurons, and so we did this by expressing a blue fluorescent protein in the tectal commissure with the yellow myelin transgene still on board. And this is a max projection image of the entire tectum, and these are just single optical sections showing these contacts like the ones I just showed you. And if we inhibit the activity of these neurons by co-expressing either a dominant negative VAMP2 or botulinum toxin to interfere with the snare machinery that underlies vesicular release, we can measure the contacts, frequency of contacts from the microglia onto these neurons, and we find that inhibiting the activity of these neurons reduces the number of contacts that the neurons receive from the microglia, implying that the microglia might be free to do something else. So when we decreased activity, we found decreased microglia neuron contacts. But is the converse true? If we were to increase activity, would microglia be spurred to contact those neurons? So we did this with a glutamate uncaging protocol, which excites nearby neurons within about 20 microns. We can elicit calcium transients in neurons. And if we do this glutamate uncaging near microglia, we find that uncaging DMSO doesn't seem to cause any change in microglia movement toward the uncaging site. But uncaging glutamate spurs microglia to move very close to the uncaging site, presumably to interact with active neurons that are excited underneath. And so we measure this as the number of movements toward the uncaging site relative to those movements on the opposite side. And of course, we can't see the neurons in this paradigm, but we also validated this chemogenetically, and activating these neurons causes microglia to contact them more frequently than non-excited neurons. So taken together, this really validates previous work that activity can bidirectionally shift the position of microglia in the tectum. But what about the consequences for myelin phagocytosis? When we inhibit neuronal activity with those same methods that I introduced previously, and then morphologically segment the myelin that's engulfed by microglia, we find that silencing neurons increases the phagocytosis of myelin by those microglia. So not only does this cause microglia to contact fewer neurons, but they instead seem to be phagocytosing more myelin. So the last part of our puzzle is to increase activity and measure myelin phagocytosis. And glutamate uncaging is great, but it only excites a few neurons and only very briefly. So we needed a way to prolongly excite neurons in the tectum for a long period of time and then study myelin phagocytosis by the whole microglia population. And so we did this with a forced swim assay in which a magnetic stir bar generates strong water currents and fish have to visually orient themselves and swim with the current. And as a visual motor task, this excites the optic tectum. And then we can measure myelin inclusions within microglia at the end of 2 days of swimming. And we found that when we segmented the myelin within these microglia, while the microglia were normal in number, size, territory, and etc., these microglia phagocytose less myelin overall. And taken together with our other data, although this is not specific to the tectum, we find that this represents a decrease in myelin phagocytosis bidirectionally modulated by activity as well. So microglia so far interact with phagocytosed myelin, perhaps in a neuronal activity regulated manner, at least in the optic tectum. But if this program really exists to refine myelination, then you might predict that in the absence of microglia, there should be an excessive myelination phenotype. And so we ablated microglia in a few different ways based on microglia development. Microglia differentiate from macrophages, which are specified from yolk sac progenitors, and we can block this specification event and we can block the differentiation event. And lastly, we can chemogenetically ablate both of these populations of cells with another transgenic driver. And with all of these approaches, we can label individual oligodendrocytes and study what myelination looks like at the single cell level in the absence of microglia. So these are individual oligodendrocytes in animals. Some have microglia, some do not. Here in pink, we have oligodendrocytes in animals in which microglia have been partially depleted by each of these 3 methods. And consistent with our results that microglia eliminate individual myelin sheaths, we find that oligodendrocytes in each of these groups that ablate microglia have a few more sheaths than normal, a few more sheaths than their control counterparts. And this is consistent with microglia eliminating sheaths. But what we weren't expecting to find is that microglia, when microglia were depleted, oligodendrocytes also formed myelin sheaths around neuronal cell bodies, these spherical structures. The cell has a few of them. And this is a phenomenon that was recently characterized by David Lyon's lab and is an ectopic myelination phenotype. We very, very seldom see this in control animals, but when microglia are depleted, we see it happen, indicating that microglia seem to protect neuronal cell bodies from being ectopically myelinated. So taken together, our working model is that microglia interact with oligodendrocytes to tune myelin targeting and protect ectopic myelination from occurring, but that microglia are attracted to active neurons, raising the possibility that this trade-off creates the regionalization of tracts, myelination of tracts throughout the nervous system. And so with that, I'd like to thank my lab. They're wonderful. Some of them are here today. Our oligodendrocyte friends on campus, my thesis committee, my graduate program, our department, and funding, and you all for your attention.
Thank you, Alexandria. That was a really great talk. We're very close to the end of your time slot, so we'll take only a couple questions perhaps. We got a first question from Igor. Great talk. What are the effects of atopic myelination?
What are the effects? Sorry, it cut out.
What are the effects of atopic myelination?
We don't know. You know, I think that if I can speculate, action potentials are generated at the axon initial segment, which is right next to the neuronal cell body. So I would imagine that ectopic myelination of cell bodies is likely to suppress action potentials. And if those neurons are myelinated and they don't die, I would imagine that would disrupt circuit function. If those neurons are recruited into circuits, they are unlikely to fire appropriately, but we don't know.
We have time for one more question. So the next question is from Dooyeon Kim. Have you depleted microglia in adult stages to see if it still affects myelination?
I have not. I would be interested to do that. The other thing that I'd be interested to do would be to raise some of these fish that had microglia depleted during these larval stages and see if there are long-term consequences for myelin in adulthood. I think that's a great question.
Great. Sorry everyone that we're skipping many of the questions. They're really great, but we just try to like keep everything under the time. So thank you, Alex. And,
feel free to reach out.
Yeah, please email Alex with any of your pressing